Well, then with that said, we are ready to begin lecture number 2. Hopefully you guys are all ready for that. All right, so just a reminder, remember the first lecture that we just completed over Introduction to Human Body, we 20 questions on our first exam here in a couple weeks. The next 20 questions will come over this lecture on cells. Fairly short lecture. We should be able to knock out a good portion of it. I don't know if we'll get it all out today, but hopefully we get a decent chunk of it done anyway, so that next week we can move on and work our way up those levels of organization. Speaking of those levels of organization, if I kind of go back to our previous lecture, we introduced those, like the second slide, and, where we went from the simplest eventually all the way to the most complex. Okay, so we've talked a little bit about cells. Okay, cells are described as a basic unit of the human body. We all begin our life as a single cell. That cell becomes 2 cells, that becomes 4 cells, and so on and so on. Here we are as adults, made up of trillions upon trillions of cells. Very, very amazing. Each cell is a living structural and functional unit enclosed by a membrane. That's kind of your textbook definition. It's living, so it performs life processes, and on the outside it's enclosed by a membrane, which we'll talk about here on the very next slide. The modern cell theory is one of the most important unifying theories in all of the field of biology, and anatomy and physiology are basically a subset of biology. So it's kind of important just have an understanding of what that cell theory is. I don't reference it much other than just right now, but it states that all organisms are composed of cells and cell products. So in other words, anything that's living is made up of cells and what the cells make. Cells are the simplest structural and functional unit of life. So, atoms technically are not living things, nor molecules. So the cell is the most basic functional unit of life. Okay. An organism's structure and functions are due to the activities of its cells. In other words, life processes occur within cells. Essentially, anything that's happening to us is because of the activity of our cells. Okay? The ability for me to speak right now is a function of cells. Contracting your muscles to take notes is because of your muscle cells. Seeing and processing what I'm putting up on the board, what I'm speaking, is courtesy of neurons and cells inside your eyes. So everything that we do is because of our cells. Cells come only from pre-existing cells. They don't just appear out of nowhere. And then cells of all species have fundamental similarities. Something that you could compare is our cells to plant cells to fungal cells. But we don't really do that. That's more of a biology class type thing. One thing that you can compare and see from species to species is that pretty much no matter what, the cells are going to be small. Okay. And there's a relationship that exists between cell size, its shape, and its function, which demonstrates a really important concept that I will reference quite a bit this semester. Okay, we talked about and defined what anatomy and physiology are the other day, right? In anatomy, we are going to study structure. Okay, in physiology, we study function. Okay, well, those two things go hand in hand. Okay, the way that we are structured, how we're built, affects how we function. Okay, and the same exists for our cells. Cells have a distinctive shape. They're built a certain way because it affects how they operate, how they function. Okay, I mentioned this the other day, but human cells are microscopic in size, meaning we can't see them with the naked eye. We need a microscope. Diameters of a cell will range from 7.5 microns or micrometers, very, very tiny, to 150 micrometers. Now, you may or may not have been exposed to micrometers before. I imagine looking at this table, you recognize at least a little bit of what you see here. What I'm kind of introducing here in this table is a look into what we call the metric system. not the kind of standard unit of measurement that is used here in America, but in most other places around the world, the metric system is used and is more commonplace, which I wish we would introduce it and just bite the bullet. Americans seems like we're just too stubborn. But it's a lot easier to do conversions and comparing sizes and weight and that kind of stuff. I don't really expect you guys to learn the metric system or anything like that, but I wanted to kind of help you visualize just what we're dealing with as far as size goes, because we will use the measurements micrometers quite a bit today. So here in America, we like to measure with inches and feet and yards, and I'm sure everybody's used a ruler or a tape measure before. Most rulers, you know, have kind of two sides to them where you can measure in inches and then flip it over, and then you can use centimeters and millimeters. Okay, centimeters are abbreviated cm, are 1/100th of a meter. A meter, you know, about a meter stick, not quite a yard. You know, I'm sure everybody knows what a football field looks like size-wise. There's 100 yards in a football field, and technically it's longer than that when you add in the end zones. But one of the most common races is the 100-yard dash. Now, 100 yards and 100 meters are not quite the same, but they're pretty close. 100 meters is a little bit longer than 100 yards. Okay, but there is 100 centimeters per meter. Okay, so that kind of gives you an idea of size-wise there. There's also 2.54 centimeters in an inch, so about 2.5 centimeters make 1 inch. Obviously, these are things visible to the eye, and you can see them very nicely on most rulers. Smaller than those are millimeters, abbreviated mm. There are 10 of those in 1 centimeter. Okay, and this is where your largest cells size-wise are in your groups of cells. Most of what we deal with today in this lecture are in our bottom 2 categories. Micrometers, looks like micrometer, but it's actually pronounced micrometers or microns. Have this kind of fancy abbreviation or symbol. Looks like the letter U, but there's a little fancier tail at the beginning of it. There are 1,000 of these guys in 1 millimeter. So they're 1/1000th of a millimeter. This is the size of most cells in your larger organelles. And then smaller than that, tremendously smaller, are nanometers, abbreviated nm. There are 1,000 of those in one of these. So very, very small. So your smallest organelles and your larger biomolecules are going to be measured in nanometers. Okay. Now, speaking of organelles and biomolecules, you can see in our illustration here of the sectional view of a cell, hopefully some stuff that you recognize from previous classes. I mentioned the other day, like the powerhouse of the cell, mitochondria. You can see essentially just how complex and amazing our cells are, with things like the powerhouse, mitochondria, rough endoplasmic reticulum, Golgi complex, the nucleus. I'm not going to expect you to worry about this figure quite yet. What we're going to do here as we roll through the lecture is basically zoom in at each of these different organelles, look at their structure and their function. So like I said, hopefully it's a nice review for a lot of you guys, whether that was just a few years ago, last semester, or whatever. Hopefully some of this stuff rings a bell. We're going to start on the outside and work our way in. So we'll start with the outer structure of a cell. It's plasma membrane. Sometimes you'll hear me just call it a cell membrane. Okay, the plasma membrane is a structure that forms the cell's outer boundary, shown here on the previous page, kind of with this kind of, I don't know, see-ish, translucent-ish blue in our picture, okay? It's the outside structure of a cell, okay? It separates the cell's internal environment from the outside environment. When I say internal environment, I mean like what's on the inside versus the external environment, our outside environment. What's outside of it, okay? Think of it like the walls of the room that you're sitting in, whether that's here in Atchison or over there in Wamego. It acts as a selective barrier and plays a role in cellular communication. When I say selective barrier, anybody have any idea what I mean? What's a barrier? It's something that, you know, keeps you from going through it, right? Okay. Well, a selective barrier chooses what can go through it. Okay. In other words, some stuff are able to pass through the plasma membrane either to come in or to go out, and it is selective and it chooses what it does, okay, or what is able to. Okay. Also plays a role in cell communication, and we'll talk a little bit more about that here in just a second. Okay. Now I compared it to the room that you're standing in, or as the walls of the room that you're standing in or sitting in, I should say. Okay. Very different though than the walls of the room that you're in that it is not really hard and solidified. Okay. You know, if I were to push out on the wall here in Atchison, it's not going to really move. Okay. Especially those outer walls that are made up of cinder blocks. Okay. That's because they're rigid structures. Okay. Plasma membrane cells have to be able to bend and change shape. Okay. And so the plasma membrane has to be very fluid and dynamic in that sense. And it is made up of fats or lipids. Fats and lipids are the same thing, and proteins that kind of float around. And the model that describes this relationship and arrangement of molecules is called the fluid mosaic model. So let's talk about the fats and the proteins that make up the basic framework for the plasma membrane. Okay, once again, lipid, same thing as a fat. Okay, looking down at the bottom of the page, we have just a very small little excerpt, little segment of the plasma membrane illustrated here, zoomed in. We see there's a set of structures that make up probably the most of it by far. And that are these things that would look like a lollipop with two sticks. Here's one zoomed in. Each of these is called a phospholipid. Okay, they make up about 75% of the plasma membrane. Very unique form of fat because they are what we call amphipathic. It's a really fancy chemical term that I'm not going to get into a whole lot on. But it just means that they have both polar or charged and non-polar or uncharged regions. I get into this a lot more in physiology. So for those of you who are in that class, you have that to look forward to. But this is unique because most fats are non-polar. Okay. And why I bring that up is because it influences the relationship that it is going to have with water. Okay. Most fats are non-polar. And non-polar structures are what we call hydrophobic. Anybody know what the suffix phobic or phobia means? Afraid, absolutely, perfect. Okay, like if you have arachnophobia, it means you're afraid of spiders. Very common fear. Okay, well, hydro means water. Okay, so the term hydrophobic literally means water fearing. That's how most fats are. If you've ever done any cooking, you've probably experienced a time where you can see like little oil drops on top of like water. Okay? Or even if you just let, you know, like do a fun experiment in your house, like with your kids, if you have kids, if you were to, you know, take a water bottle, put some water in there, and then dump some oil in there, you shake it up, eventually that oil is going to sit on top of that water. It's not going to ever mix in. And that's because oil is just liquid fat. Okay. Well, the tails of a phospholipid, these little two squiggly lines here, are going to be just like any other fat in that they don't like water. Okay, they are hydrophobic. Okay, well, the heads are hydrophilic. Hydrophilic means that they actually like water and they're attracted to it. Okay, and they're going to seek it out. Okay, Well, in our body, we have a whole bunch of these phospholipids because we have a whole bunch of cells, and they're the major structures that form the plasma membrane of cells. And what happens is something really cool. You have a cluster of phospholipids. They're going to organize themselves into a bilayer. And we call it a bilayer because there are two layers. Bi means two, right? I'm sure that's pretty common prefix you've come across before. If you have a bicycle, it's because it's got two wheels. Okay. What you see is the polar heads are pointing in opposite directions, whereas the tails are pointing together. And the reason that the polar heads point in opposite directions is because on the inside of the cell is a fluid that we're going to talk about here in just a second called cytosol, which is predominantly water. And on the outside of the cell is a fluid called extracellular fluid, which is predominantly water. So that the heads point where the water is, the tails point away from the water in the center. And that accounts for a big reason why cells have this selective, or the plasma membrane has this ability to act as a selective barrier. Okay. Other fats that are found within the plasma membrane include glycolipids. Now you see the prefix glyco a couple different times here in this figure at the bottom. Glyco means sugar or carbohydrate. Those terms actually mean the same thing, okay, because sugar is already a form of carbohydrate. But a glycolipid, therefore, is a carbohydrate lipid kind of hybrid, okay? They're not a whole prominent, or not very prominent, I should say. They're only about 5% of the lipids in the plasma membrane. We only find them facing the extracellular space, which means they're only facing outward from the cell. But they help with cell adhesion, so like cells sticking together. They also help with mediating cell-to-cell recognition and communication, so cells can recognize and communicate with one another. And they help with cell growth and development. Okay. And as I mentioned, they're lipids with carbohydrate groups attached. Cholesterol, that's a term you've probably heard of before. Something that, you know, you might just immediately think, oh no, this is a bad thing, because people talk about their cholesterol levels all the time. They're talking about the cholesterol that's floating around in their blood, something that we don't want a whole bunch of. But we naturally and absolutely should have some cholesterol because it is fairly prominent, about 20% of the fats in our plasma membrane. You can see the little yellow squiggly lines here. They are found kind of in the center of the plasma membrane, and they help actually strengthen it. But they also at the same time do decrease its flexibility. Other prominent structures within the plasma membrane are proteins, which are categorized as either integral or peripheral based on where they're at within the membrane. Okay, integral proteins, just think of them, they travel throughout the interior of it, so they're found not just on the outside or the inside, but also within the middle. Most of them are transmembrane. There's that prefix trans again. Trans means across or through, and so that means they, you know, go through all the way, touching the outside and the inside of the plasma membrane. Okay, so protruding into the cytosol and extracellular fluid. Now, many of our integral proteins are glycoproteins, like this one here, which is just like a glycolipid in that it's got a carbohydrate attached to it. Okay, and together glycoproteins and glycolipids are going to give each cell a pretty unique kind of exterior coat. Okay, this sugary coat is called the glycocalyx, and this pattern of carbohydrates varies from cell to cell, which enables it to basically tell cells that they're different from one another, or it enables cells to recognize one another. Okay, it'd be like, you know, looking around the room and we're all wearing a unique, you know, set of clothing, right? Well, cells wear their own unique clothing all the time, and that enables them to recognize one another. A good example of why this is important. So, you know, obviously we're dealing with the pandemic right now, and we talk about immunity and developing resistance to things like viruses and bacteria. Well, bacteria have their own coat, okay? And our immune system cells recognize, hopefully anyway, when a foreign cell like a bacteria has come into the body. Okay? And one thing we don't want is our immune system cells recognizing our own body cells as foreign and attacking them. Okay? So that's very, very important. Other examples why it would be important, it like prevents protein or protects cells from being digested by enzymes that are found in our extracellular fluid. Okay. Other proteins include peripheral proteins. You find them either just on the outside or the inside, like what I'm showing you down here. Here's some peripherals on the out, or one peripheral anyway. They're not embedded in the membrane, only attached to the inner or outer surface. As far as the kind of three images, I'm not really worried about you knowing this one or this one. Actually, this is the one I want you to know. Highlight it, whatever. Where it says polar head, non-polar tails, it might not be a bad idea to label. You know, the head, remember, is hydrophilic. The tails are hydrophobic. So if you need to write that in, might not be a bad idea. All right. One last little slide here on those lipids and proteins. So that's how we kind of classify them structurally. We can also classify our different lipids and proteins based on what they do for our cells, their function, in other words. Okay. Some integral proteins form ion channels, pores, or holes that allow specific ions to flow through to get into or out of the cells. Okay. Once again, kind of using the room that you're in as a kind of analogy, pretend that the room that you're in is a cell, okay, and you were able to come in or out of the room thanks to a doorway, right? Okay, that's what ion channels, pores, or holes, I usually call them channels, allow for. Okay, now most of these are selective, meaning that they only allow a single type of ion to pass through. So in other words, you know, this channel here, or pore, whatever you want to call it, maybe it's going to allow sodium. Maybe these are sodium ions. Okay. It's not going to allow potassium or chloride or another ion through. It's only going to allow a certain ion through. Does that make sense? Okay. Now, very similar to channels are carriers. Carriers also go by the term transporters. They move something or carry it, hence the name, across the plasma membrane from one side to the other. They also are going to work selectively. This is, for example, how we get glucose in and out of our cells. The difference, it looks like in this illustration here that there are two different proteins, right? Two different carriers. Well, it's actually the same carrier in one position, and then here it's showing it that it's changing its shape. Okay, so the key here is it changes a physical shape, where at one point it's more open to the outside of the cell to allow the substance to bind to it, and then it changes its shape and the opening allows for that substance, whatever that is, to be spit out, or I guess spit into the cell. So when you came into your classroom, were you using something more like a channel or a carrier? Channel, okay? And if you didn't catch it, I said when you came into the room, you used a doorway. Okay? I didn't say door, okay? You use the doorway, like the door frame around the door, okay? Channels can have a gate or a door that closes the opening. But when you came into the room, the shape of the door frame, the doorway did not change, okay? So it's not a physical change like what happens with carriers. Okay. Receptors are integral proteins that serve as cellular recognition sites. Each recognizes and binds a specific type of molecule known as a ligand. This is, for example, how a hormone that we release in one part of the body can target one specific cell or one specific organ. For example, we release a hormone, and I've got antidiuretic listed here, but I'll use thyroid stimulating hormone. Thyroid stimulating hormone is released in our brain. Guess what organ it stimulates? The thyroid gland. Well, how does the thyroid gland get acted on? Well, the cells of the thyroid gland have receptors for that hormone. No other organ should have those same receptors. Does that make sense? Okay, enzymes are integral or peripheral proteins that catalyze or speed up chemical reactions at the inside or the outside of the plasma membrane. So they just help with chemical reaction processes. Linkers, kind of common sense, I would assume by their name, they help link or anchor proteins to one another. So they help with like cells connecting. Um, and then membrane glycoproteins, glycolipids. This is what I was talking about with that sugary coat on the previous page. And they enable cells to recognize one another. Very important, like when tissues are forming, and when like our immune system cells respond to potentially dangerous foreign cells. As far as the images, I'm not really worried about you knowing those. Just know what the different categories of their proteins are. So that's the plasma membrane and the structures associated with it. You guys don't have these three things to fill in, do you? Okay, it's these, I think, right? Let's talk about stuff on the inside of the cell now. All right, inside of our cells, we have a gel-like substance called cytoplasm. It is made up of the various organelles, all those little machines and molecules suspended in a semi-fluid substance called cytosol, or what we will also refer to as the intracellular fluid. Okay, so we've already been talking about the cytosol. Cytosol, I pointed out a couple slides ago, is just the fluid on the inside of our cells. Another term for it, as you can see and already said, intracellular fluid. You'll see this prefix intra quite a bit this semester. Intra means inside or within, so intracellular means within the cell. Okay, I'll be honest with you, I kind of use cytosol and cytoplasm interchangeably. Technically, that's not a perfect way to do it because it's not accurate, but I kind of catch myself doing that from time to time. Cytosol is the fluid. Cytoplasm is the fluid plus the organelles. So that's why it's not just kind of a watery thing. We call it a gel-like substance, okay, because those organelles kind of thicken it up, okay. Now, the cytosol of human cells is far from being a structureless fluid. It's permeated by an intricate three-dimensional array of interconnected framework called the cytoskeleton, okay. So we know cyto means cell. Skeleton, obviously, we all know what that is, our bones. Okay, well, our cells don't have bones, but they've got these proteins that kind of help give our cells their shape. It gives organization to our cells, kind of like what our bones do for our whole total body. Okay, there are three components to the cytoskeleton: microtubules, intermediate fibers, or intermediate filaments, as you can see, they are also described as, and then microfilaments. Okay, we're going to go from the biggest to the smallest. So microtubules are the largest. They are hollow cylinders with an outer diameter of 25 nanometers. So they're a cylinder. Think of them kind of like a tube or a pipe. You know, I'm sure you've all seen PVC pipes, drainage pipes, that kind of stuff. That's kind of their shape. They've got a rigid outside and then a hollow internal structure to them, made up of, or have a diameter of 25 nanometers. They consist of longitudinal arrays of the protein tubulin. Each of these little blue dots is a protein called tubulin. So I don't know where that name comes from, but it's kind of handy. Tubulin makes microtubules, and they have a tube shape. Okay, they begin in organelles that we'll talk about on the next page called centrosomes. Not really too concerned with most of this information. These four things here are really what I want you to know. What do they do for us? In other words, they help form the shape of a cell. They help with moving organelles. What they're probably best known for and seen most for doing is number 3, aid in the movement of chromosomes during cell division, and they help with forming specialized cell projections. I'm going to talk more about these here on this bullet. If you've ever taken a biology class and you've learned about mitosis and meiosis when cells divide, okay, you can actually see what I'm kind of going to refer to. There's a period when a cell gets ready to divide, and you see these kind of long, spindly looking things start moving around the cell. It always looks to me like spider legs. And this kind of looks like a spider. Okay. Each of these long leg-like structures is a microtubule. And they are what grab and pull the chromosomes apart when a cell divides. So if you've seen that, you kind of can visualize it. Microtubules are what are grabbing those chromosomes. Intermediate fibers or intermediate filaments, same thing, thicker than microfilaments, than in microtubules. They don't have a special type of protein that makes them. It actually varies from cell to cell, but their function is to resist mechanical stress to the cell, stabilize the position of organelles, and help cells attach to one another. The smallest, thinnest ones are our microfilaments, very small, only having a diameter of about 7 nanometers, made up of a protein called actin. They help generate movement, like when our muscles contract, when our cells divide, or when our cells move. That's what locomotion means, which we'll talk about on the next page. They provide for much of the mechanical support responsible for basic strength and shape of the cells. Notice, the kind of purple squiggly lines here, I guess maybe purple, pinkish, whatever. It's on the outside, so it's kind of pushing out on that plasma membrane and gives shape to the cell. And they provide support for cellular extensions like this one here called a microvillus, which we're going to talk about on the next page. Okay. As far as those images on this page, don't worry about them, just the information. All right, centrosomes, which I was just talking a little bit about with regards to microtubules, because that's where they begin. Centrosomes are organelles located very near the nucleus. Structurally, they consist of two centrioles and pericentriolar material. You can see those things are labeled over here. Centrioles are cylindrical structures composed of clusters of microtubules. Even though they're very small organelles, they're very complicated, three-dimensionally, lots going on with them. Not really concerned too much about those two bullets. These are the three things I definitely want you to know, which is what they do. They help anchor and assemble microtubules, which we kind of already just talked about. Therefore, that also helps with moving cells and moving chromosomes during cell division. And they also help with moving flagella and cilia, which we're going to talk about. And you guys just fill those blanks in. So we'll do that here in just a second. Okay. Now, along with the cytoskeleton and the plasma membrane, our cells can have this ability to have some pretty unique kind of external features, extending the plasma membrane outward, forming these tiny finger-like processes. So in other words, cells don't always have kind of just a nice round circular shape or a nice square rectangular shape. In some cases, they might have a side that projects outward, like shown here with what we call our microvilli. Now you can see the term microvilli here, whereas in our picture, it's labeled microvillus. Villus is singular, and you can see only one is circled. Villi is plural, okay, but it's the same thing, okay. But these are found in epithelial cells that line our intestines and other areas where absorption is very important, okay. Absorption is where something comes through the plasma membrane into the cell, okay. They are about 1 to 2 microns in length and can increase the surface area of a cell about 20-fold. You're going to hear this phrase surface area quite a bit this semester. Think of surface area as like the amount of contact or the amount of something a cell has. In this case, the amount of plasma membrane this cell has is increased tremendously, okay? Let me just kind of explain it this way, okay? So everybody agrees with me, I would assume anyway, that the distance from here to here is the same on this side of the cell as it is from here to here on this side of the cell. Okay, but the amount of blue that you see here, the amount of plasma membrane, very thin, but it's not nearly as much as what you have here because of these finger-like extensions. Okay, for absorption to occur down here, you only have from here to here for it to occur. Okay, this is where you'd find those channels and those carriers that we just talked about. Not as much or not as many of those channels or carriers could be there as what you could have here. Okay, not only do you have them in one row, but as you can see, since cells are three-dimensionally shaped, you have several rows of them, which means there's a great capacity to have those channels, thus a great ability to carry stuff into the cell. Make sense? Okay. So, like I said, we have them in places like our small intestines, our kidneys. That's where we do a lot of exchanging. Like your small intestines, where most of the stuff that we eat and drink is absorbed. Here's a picture of some in a real cell. This is zoomed in under a special microscope 5,000 times, which will make it I can see. You can also see in both pictures they label that microfilaments, those little proteins that we just talked about, cytoskeleton structures, are going to be what help to form them. Cilia are hair-like processes about 7 to 10 microns in length, composed of microtubules. They help move in a rhythmic, coordinated way to push substances along the surface of the cell. Okay, they look like tiny little hairs. Here we can see 3,000 times with the naked eye could see. But they move in a very coordinated manner, like what you show here. Cilium is the same thing, like we saw above, is singular, cilia is plural. But the key is they don't move the cell. The cell stays put, but they kind of act like a conveyor belt and they push something along the surface of the cell. We have these in a lot of different places. Our respiratory tract, our airways on the way into the lungs. This is what helps like keep stuff from, collecting in our airways. The cilia helps push stuff up. When we breathe in dust and particles and that kind of stuff, there's mucus on the inside of our airways that act as a glue trap, and then the mucus and the other gunk gets pushed upwards into our throat, and then we either kind of hack it out, cough it out, or it gets swallowed into our stomach. Cilia are, like I said, found all over the place. The fallopian tubes of a female, the cilia there helps to move the egg after it's been ovulated. It's kind of a cool example. We only have one example in our body where there is a cell with a flagellum. Flagellum singular, flagella plural, long, fine thread-like organelles that protrude from the cell surface, usually move an entire cell. So different from cilia in that the cell is actually able to move. This is what enables sperm cells to swim in a rapidly wiggling wave-like pattern. You've all probably seen videos of sperm under a microscope swimming like a snake. And this is what enables the sperm to eventually make its way up into those fallopian tubes to hopefully find an ovulated egg. As far as any of the pictures on this page go, don't worry about them. Just the info. All right, let's talk about the nucleus. This is our largest organelle in our cells. And within the nucleus are most of the cell's hereditary units called genes that control cell structure and cell activities. Notice genes with a G, not with a J. It's not the type that you wear. But genes are what control the ability of the cell to have its structure and kind of really more importantly is its activity. Okay, genes are what give us everything that's unique about ourselves. You look around the room at each other, whether you're there in Wamego or here in Atchison, and we can see things like our hair color, our skin color, our eye color, okay? Those different physical traits are because of the genes that we've inherited from our parents, okay? Well, I was just mentioning earlier the concept of mitosis. When cells divide, chromosomes are pulled apart. So a chromosome is a structure that contains thousands of genes, okay? Human cells typically contain 46 chromosomes, 23 that we inherit from each parent, okay? When the sperm from dad and the egg unite from mom, okay, 23 chromosomes are carried in the sperm, 23 are carried in the egg, and 23 plus 23 come together and form 46. 46 chromosomes are found in that original zygote cell, okay? A chromosome is a long, winding molecule of DNA. Okay, here's some really cool pictures. This is zoomed in under a microscope over 6,000 times to show you kind of that capital X shape to them. But basically, if you kind of just, were to highlight one little segment of it, okay, that would be like where you'd find one gene. Okay, maybe that's the gene that color or gives you your hair color. Okay, over here, this would be another gene. And, like I said, there's thousands of genes on one singular chromosome. Okay. Now, what we see most of the time, like what we show down here, this is a zoom in of a nucleus, is that cells don't always have their DNA organized into chromosomes. Okay. Only before a cell gets ready to divide do the chromosomes appear. Okay, that's when the DNA is winding up into this kind of special shape. That way, when the cells do divide, it's easy for the division to occur, all the DNA to go into the correct cell. Okay, most of the time you find the DNA in an unwound state in what we call chromatin. Okay, chromatin is not just DNA, but it's also proteins. And I was just, tell students, think of RNA like DNA's cousins. Okay, it looks kind of like a grainy mass. Okay, we see it here in our real picture. We can also see it over here in our illustrated version. Okay, and this tells us that the cell's not ready for division. Okay, because it's more than likely just doing normal cell things. Okay. Now, the structure of the nucleus includes a nucleoplasm, a nucleolus, a nuclear envelope, nuclear pores. Okay, the nucleoplasm is essentially the same thing as the cytoplasm that we just talked about. It's the semi-fluid on the inside. So it's not labeled, but this is just the fluid here where that chromatin is. That would be the nucleoplasm. Okay, like our cytoplasm, it's enclosed on the inside of our plasma membrane. Well, our nucleus has its own kind of like plasma membrane, what we call the nuclear envelope. Sometimes you'll actually even just hear me call it the nuclear membrane. Just like the plasma membrane that we just talked about, it's got two layers. It's a double layered structure that prevents certain things from coming in and out of the nucleus. Okay, and just like our plasma membrane, there are doorways to allow substances to come through it, okay, in or out. Each of those is called a nuclear pore. Pretty complicated little structures, but they're very important for controlling movement of substances between the nucleus and the cytoplasm. Okay. Now, I haven't talked about this yet, but towards the center of the nucleus is almost like a separate organelle. Okay, it's called the nucleolus. The nucleolus is a small non-membranous body within the nucleus. So in other words, it doesn't have a membrane around it. But its job is to synthesize RNA, which is then going to combine with proteins to form subunits that we will call ribosomes that we're going to talk about on the next page. Okay. And that's what these little dots are. But essentially, the nucleolus is where we make RNA. Now, I just kind of described DNA and RNA's relationship like they're cousins. Okay, you may recognize that term RNA, especially here in the last year or so, if you paid attention to what's going on with vaccinations and that kind of stuff, because the vaccines are mRNA based. You guys heard that phrase before. Okay. What the vaccine is, it's not the virus, it's not a protein, it's RNA, which is something our body naturally makes. Okay. Well, viruses have RNA. Okay. And so when a person gets a vaccine, they're kind of getting a building block that our body normally makes, that a virus normally makes. So it's kind of cool. Very, very new form of vaccination, kind of very different than what's usually done or previously done anyway. Okay, so what does your nucleus do for you? It controls all cell activities because it contains the DNA that has the instructions. The DNA is used to make the RNA. RNA is then used to make the proteins. Proteins do things like give us our skin color, hair color, eye color. So that's how cell activity is controlled. We store our DNA and other genetic information in our nucleus, and we synthesize more DNA, and as I've already said, RNA in the nucleus. So those are kind of the three major things. As far as the images on this page, don't worry about these two figures. I always like to try to put real pictures in whenever I can, but rarely do I use those on exams because they're kind of harder to tell things apart. I do want you to be able to identify figure A and B here on the bottom left, though, because there are some very important things. Okay, let's talk about those ribosomes that I was just kind of referencing, along with another couple organelles that collectively we call the endoplasmic reticulum. Okay, ribosomes are the sites of protein synthesis. If you don't know what the word synthesis means, it just means like creation, formation, building, whatever. Okay, so in other words, ribosomes are the protein factories of our cell. Very tiny little organelles, but they're very complex, structurally made up of more than 50 proteins. Another type of RNA, not the mRNA that I just mentioned, but ribosomal RNA, or RNA for short. There's two subunits that make them up, one large and one small. Really not a whole lot that I want to focus on there. But they're found in all sorts of different places inside of our cell. They're typically found on the outer surface of nuclear membrane or envelope to a folded membrane known as the endoplasmic reticulum. In fact, if we go back to this previous page, each of these little white dots that you see in the picture on the outside of the nuclear envelope is a ribosome. You also find them attached to the endoplasmic reticulum, or specifically the rough endoplasmic reticulum. There's also ribosomes that are just kind of free floating around inside the cytoplasm. We just call those free ribosomes for that reason. There's also ribosomes inside our mitochondria, so we'll call those mitochondrial ribosomes. All right, the endoplasmic reticulum, or ER for short, not emergency room, but endoplasmic reticulum is a network of membranes extending throughout the cytoplasm. It consists of tubular membranes and flattened sacs called cisternae that are interconnected. There's an internal space enclosed by the membrane called the lumen. There's that word lumen that I introduced to you guys on Tuesday. Lumen, we said, was the hollow part of an organ. In this case, it's the hollow part of an organelle. The ER is continuous with the outer membrane of the nuclear envelope, and therefore the space between the two nuclear membranes is the same as the lumen of the ER. Pictured here at the bottom, we've got a couple of different things. A real view here under a special type of microscope, 45,000 times what our naked eye could see, to show you the two forms, rough and smooth. Okay, but this is the illustration I'll kind of reference the most, showing you those two forms, rough and smooth. And you can see their connection in relationship to the nuclear envelope, and we can see little dots are now labeled, although they weren't on the previous page. We now see those are ribosomes. Okay. Now, if I zoom in, you can see they are these flattened membrane structures. The very kind of thin gold line that you can see here would be the cisternae, and then the space where we can see kind of these little whitish dots is the lumen. And we see kind of a similar situation here for our illustration of the smooth ER. Difference between the rough and the smooth is simple as, are there ribosomes or not? Okay. The rough appears rough because of the ribosomes that are attached. And as we just learned, ribosomes are what produce the proteins. So the main function of the RER is to synthesize proteins. Okay. The smooth ER has no ribosomes. It has no role, therefore, with producing and synthesizing our proteins. Instead, this is where our fats come from. Things like fatty acids, steroid hormones or steroids are a form of fat. Cholesterol is another form of fat, as we've already kind of talked this morning. SCR is also responsible for inactivating and detoxifying potentially harmful substances, things like drugs, alcohol, pesticides. You guys know what the term carcinogen means? It's a fancy term for like a chemical that causes cancer. So obviously not a good thing. Things that we don't want in our body, but we become exposed to. So your SCR cleans you up, basically, and it cleans up the cells, especially on our liver. Okay. SCRs also involve carb metabolism and calcium storage. And in a few weeks, we're going to talk about the SCR in our muscle cells, where we actually will change its name and called the sarcoplasmic reticulum. But it is responsible for releasing calcium ions that help trigger muscular contractions. Okay. Now, what's really cool about the making of proteins, the making of fats, if it's down here for the SCR, those proteins in the RER, the fats here from the SCR, will accumulate on the inside of the ER, like these little white dots that I've already kind of referenced. Those, in this case, are probably proteins that are being made. Okay. You kind of zoom in a little bit. You see how this is kind of forming into a ball? Okay. The proteins that are made here and the fats that are made down here need to be moved and carried from the ER to the next organelle that we're going to talk about on the next slide. So what happens is those proteins or fats will accumulate on the inside, and then a pretty cool process occurs where the membranes kind of pinch inward and a bubble breaks free. Okay. That bubble is called a transport vesicle. You kind of think instead of vesicle, called a transport vehicle, because a vehicle transports something, right? Okay. Well, those bubbles, those vesicles, carry those proteins and the fats from the SCR to the Golgi complex, Golgi body, Golgi apparatus, okay, which we're going to talk about in just a second. Make sure you can identify those different 4 organelles that are labeled in this picture, along with that information at the top. And now we can talk about the Golgi, which is what I usually call it. You can call it Golgi body, Golgi apparatus, Golgi complex. I just call it Golgi for short. Okay. This is a similarly structured organelle. You see kind of a flattened stack of membranes. To me, it kind of looks like a stack of pancakes. There's hollow spaces on the inside, just like what we saw with the ER. This is where those proteins and those fats are carried once they've left the ER. In this case, we show rough ER, these green blobs coming here. Okay. The key function of the Golgi is to package different molecules together that can be used elsewhere in the cellular body. I tell students to think of these like the UPS, the FedEx, the postal service of your cell. They receive and ship packages. Okay, the packages being proteins and fats. Okay, now there's distinctive parts to the Golgi. Okay, the entry or cis face is the side of the Golgi that is facing the RER and the SER. In this case, it's the RER. This is where the package comes. Okay, so this is like the UPS store where you take a package and drop it off, right? Okay. Once you drop off the package, if you're sending, grandma care package or something like that, okay, your job's done, and then it's all in their hands. Okay, they're gonna, slap some barcodes on there, some tracking numbers, that kind of stuff. That's kind of what the medial part of the Golgi does. The medial cisterna allows for processing. What happens is sometimes proteins need to be altered. Like earlier in the lecture, we talked about glycoproteins, glycolipids. Okay, well, those aren't immediately made. We have to attach the carbohydrate to the protein or the carbohydrate to the fat. This is going to be where that a lot of times takes place. Okay, and then once the like the package has got the barcodes and all that stuff on it, it's ready to be shipped and delivered, right? Well, that's what the exit or transphase is able to do. Okay, this is where we kind of do the same thing. Those bubbles form and break free. Okay, enabling the protein, the glycoprotein, the fat to be delivered. But where it gets delivered is dependent upon what it is. Okay, like this illustration down here at the bottom is going to show some of those proteins are going to get just dumped out of the cell altogether, like we show right here. What if it's a protein that's going to become a channel? Maybe it's going to have to become part of the plasma membrane so it can merge with the plasma membrane. Okay. In some cases, we form a whole new organelle called a lysosome. And we're going to talk about lysosomes here on the very next slide. Okay. So this is a really cool picture to kind of show you that relationship tracing back to the ribosomes and the RER, and then eventually the ultimate destination of those proteins or fats. Here's a a real deal view of it. You can see the vesicles forming, shown there in green. Another real deal view of it. But this is actually the only one I really want you to know. Not my favorite picture, but go ahead and highlight it in case I do decide to put it on the exam. All right, let's talk about lysosomes and a couple other organelles. All right, lysosomes, organelles about 1/2 to 1 micron in diameter, surrounded by a single membrane, formed from the Golgi complex like we just showed. Okay, so there's not 2 parts to the membrane, it's just a single membrane. But inside of them are very potent hydrolytic digestive enzymes. Hydrolytic means that they can just like break things down. Okay, very, very strong. I always tell students, think of these like the garbage disposals of the cell because of these various enzymes that enable them to digest certain things. Things that they can break down or hydrolyze would be our proteins, our nucleic acids. That's what DNA and RNA are, complex carbohydrates and fats. So any of the organic compounds in a cell. They can actually digest and dispose any worn out organelle. So like when an organelle like starts to kind of break down, they digest and dispose of those. That process is called autophagy. Auto is a prefix that means self, and phagy or phage means to eat. So it kind of translates to mean self-eat. They can degrade foreign material brought into the cell. This is what our white blood cells, our immune system cells, that I always tell students, think of those like Pac-Man, they gobble stuff up. Once those viruses, bacteria are gobbled up, then the lysosomes digest and kill the bacteria or the virus. Okay, lysosomes can actually destroy the entire cell. It's kind of like hitting the self-destruct button. That process is called autolysis, and that's something that needs to happen from time to time. Okay, down at the bottom we can show a couple different illustrations, and then one singular illustration I should say, and then a couple pictures to show them off under a special microscope. Not really too concerned with any of the pictures on this page though. Peroxisomes look like lysosomes in structure. They're actually smaller and they carry out different functions. We have a lot of them in our liver cells. They degrade hydrogen peroxide, which is potentially toxic to our body. I'm sure you all have heard of hydrogen peroxide before. You can actually buy that over the counter commonly used to like treat wounds and that kind of stuff. You wouldn't want that just like floating around in your body. It is something we actually do produce, and peroxisomes help to neutralize it. They also neutralize free radicals, which are dangerous molecules that actually are really well known for causing cancer. So peroxisomes, in a sense, help prevent cancer. Peroxisomes also detoxify drugs and damaging substances. So they're the second organelle. Remember, smooth ER is able to help detoxify drugs and damaging substances, and then they can decompose a type of fat. It's called fatty acids. Proteasomes, there's not a lot of information about them, and actually, I'll be honest, I don't ever remember learning about these when I was in school. I took a lot of classes where we learned about cells. I don't know if that means they're kind of just recently discovered or just so minuscule that most textbooks don't include them. But they're barrel-shaped organelles that destroy unneeded damage or faulty proteins by cutting those proteins into smaller peptides, which is like smaller segments. So, okay, our last organelle will be the powerhouses of our cell, our mitochondria. It says production, but it's energy production. Sorry, it's kind of cut off one side. Okay, these are organelles that are the site of a process called aerobic respiration. Now, you may maybe seen the term aerobic. You might have heard the term anaerobic before, especially if you're interested in exercise. When it comes to this class, aerobic just means with oxygen. Okay, respiration refers to the production of energy, and that's what they do. That's why we call them the powerhouses in the cell. Very common phrase you hear when I ask students sometimes what that means. They give me that blank stare. Well, now you know. Okay. The whole reason that we breathe in oxygen is to provide that oxygen to our mitochondria so that they can provide us with energy. Okay. The more active a cell is, as in like our muscle cells, the more energy they require, the more mitochondria they require. Okay. Now the term mitochondria is plural, mitochondrion is singular. And like we've seen with several other organelles, they have a double membrane. Okay, what we call the outer mitochondrial membrane, shown here in kind of the more darker orange, bloodish orange color. And then the kind of lighter orange color shows you the inner mitochondrial membrane. I really like this image because it's got it opened up and exposed to show that the inner mitochondrial membrane is folded inward on itself. Those inward foldings of that inner membrane have a special name, what we call the cristae or cristae, however you want to pronounce that. Okay, like what we talked about earlier with those little finger-like extensions called microvilli, cristae increase its surface area tremendously. Okay, here we can see real pictures of mitochondria, okay, or mitochondria, zoomed in tremendously here to show you those foldings. Okay. What we see is if we zoom in out a little segment of that folding, each of these little orange dots is an enzyme. These are proteins that are helpful in the making of the ATP, the energy. ATP is energy. Okay. If you've taken classes, which I imagine many of you have, where you learned about glycolysis, Krebs cycle, electron transport chain, any of those things sound familiar to you, the Krebs cycle, or excuse me, the electron transport chain takes place along the cristae. It's the enzymes actually involved that help to make the ATP. Now, inside of the inner membrane, inside those cristae, is a semi-fluid, what we call matrix. Okay, the matrix is home to things like the ribosomes that you see here. It's also home to a special type of DNA, mitochondrial DNA, It's the only other organelle that has its own DNA aside from the nucleus. This is where the Krebs cycle takes place. If you've learned about that, now you know where that takes place. Okay. As far as the images go, I do want you to know kind of these two. Really, this one is the one that I want you to know. Don't really worry about enzymes, just the ones that are bolded there for you. But now you know, hopefully, why we call it the powerhouse of the cell. And with that said, we have talked about all of the major organelles of a cell. So to kind of wrap things up, this is a nice transitional slide to move us into the lecture that will start on Tuesday. We'll do that today because we're almost out of time. But we're moving up the ladder on our levels of organization, and we'll climb the ladder to the tissue level on Tuesday. So this is a nice slide to transition us to that point. Our body is going to have nearly 100 trillion cells. Like I said, trillions upon trillions of cells. Very amazing considering we started off our life as one single cell. But of those 100 trillion cells, there's only about 200 different types of cells. So 200 different types is not a whole lot when you compare that to the 100 trillion that we have, roughly 100 trillion. Now, of those 200 different cell types, they are going to vary in size and in their shape. Okay. Largest cells, what we call a oocyte or egg cell, have a diameter of about 140 microns, which is barely visible to your naked eye. You can actually see those without a microscope. Whereas red blood cells, your smallest cells, have a diameter of 8 microns. They're actually the most numerous, and they're the reason why our blood appears red. Okay. Shape-wise and kind of structurally, you can see we've got a few different examples of cells here, how unique they are from one another. Some are round, oval, flat, column-shaped, cube-shaped, elongated, star-shaped, cylindrical, or disc-shaped. Okay. Some will have extension, like the microvilli that we talked about, the flagella that we talked about. Some have things like these dendrites. Okay. Once again, I mentioned this earlier in the lecture, but very important theme in this class is the relationship that exists between shape and function, and that is something that's not just important at the macro level, at the whole body level, but at the cellular level. So a cell shape is directly related to its function in the body. And as you will see in our next lecture, cell diversity permits the organization of cells into more complex tissues and organs. So we'll get to that point next week. So for now, we'll just call it a day. Nothing picture-wise on this page that you need to worry about for the exam. Just a good way to illustrate those different shapes and sizes and that kind of stuff. So